The DNA ladder is twisted up tight and then wound tightly onto spools and then into bundles called chromosomes. Now, these chromosomes are big enough to see under a microscope. All the DNA in your body could fit in a teaspoon. Yet, if you were to untwist and unwind all the DNA in your body and stretch it out end to end, you'd die. But your DNA joined end to end would reach to the moon and back some 1,500 times. That's some pretty impressive programming. Microsoft could take some lessons from there. Now, the great apes have 24 pairs of chromosomes, but humans only have 23 pair. But it is assumed that humans descended from the great apes. It was claimed then that two chromosomes fused together into one at some time in the past as humans were evolving. It was believed that human chromosome number 2 was the resultant chromosome composed of 2 fused ape chromosomes. We discussed this claim previously on several shows, as well as Crevo Rant number 181, and how the chromosomal fusion argument is bust. More news has come out since the airing of these shows and our previous interview with Dr. Jeffrey Tomkins, who joins me again. Dr. Tomkins' PhD is in genetics with a master's in plant science and a bachelor's in agriculture education, and he was on faculty in the Department of Genetics and Biochemistry at Clemson University for a decade. He also ran his own genomics institute for 5 years. He is now a research scientist with the Institute for Creation Research, where he continues to publish various papers. Some of those papers we're going to discuss today. He joins me from his office in Dallas. Welcome to the show again, Dr. Tomkins.
Thank you. Good to be back.
Now let's start off. I want to backtrack to a paper you came out with in 2011 addressing this claim that humans have a fused chromosome, which allegedly supports evolutionary claims.
Right.
So let's go back in time for a minute, back to 2006, and revisit a lecture given by evolutionist Dr. Kenneth Miller at Case Western University. The lecture is entitled The Collapse of Intelligent Design. Now, it's interesting to note he very confidently claims that not only is there evidence that humans had 2 chromosomes fused, but that this is such powerful proof of evolution that he brought it up in the Kitzmiller-Dover trial, a court case which frankly wasn't a court case, but a 3-ring circus complete with a circus judge. A circuit judge. The segment of Miller's lecture where he discusses the chromosome fusion was linked to all over the internet. In fact, even today, I get comments from atheists and skeptics pointing me to that segment posted on YouTube under the provocative title, How to Shut Up Pesky Creationists. They hand me this video as if I haven't seen it before, as if it's absolute proof of evolution. I'd like to play a segment of that lecture so we can hear straight from the horse's mouth exactly what Miller said.
What must have happened is that one pair of chromosomes must have gotten fused. So we should be able to look at our genome and discover that one of our chromosomes resulted from the fusion of 2 primate chromosomes. So we should be able to look around our genome. And you know what? If we don't find it, evolution is wrong. We don't share a common ancestor.
Okay, so in 2011, you and Dr. Jerry Bergman came out with a paper showing that this alleged fusion site was small, highly degenerate, and really not much of a fusion signature.
Right.
2 years later, you published a second paper showing that the fusion site actually contained the genetic code for an important feature inside a highly expressed, life-supporting gene. Now, we discussed this when you were on the show last, and I got a firestorm of emails and comments saying that you were wrong, and even Kevin Miller said you were wrong. So clearly you were wrong.
Okay, well, should we backtrack and
Sure.
and cover the details of that discovery? Okay. Well, the fusion site actually is a functional feature inside an RNA helicase long non-coding RNA gene. So this gene produces functional long non-coding RNAs of various sizes. Now this gene has 3 exons or 3 coding regions. The other areas of the messenger RNAs are spliced out of the transcripts made from this gene. So you get short variants and you get long variants of this gene. So Ken Miller actually gave an interview to a reporter that works for, what was that magazine? I've got it here, Mother Jones.
Oh, yes.
Magazine. Okay. Does that sound familiar?
Yes,
okay.
It does, yes.
So the reporter was a guy named Mooney. So Mooney basically got the interview from Ken Miller. So Ken Miller says that the fusion site is actually 1,300 bases away from the gene.
Right.
So a friend of mine at the Discovery Institute said, Jeff, did you see this article? And I said, no, I didn't. He said, well, this is what Miller is saying. He said, you need to contact him and get this thing settled. He said, Ken Miller never responds to me whenever I send him an email, but maybe he'll respond to you. So I sent Ken Miller an email, and he actually responded within an hour. Very quick. And he was very cordial. He gave a nice long email explaining his position. But the interesting thing was he obviously didn't read my paper, because if he would have read my paper in its entirety, he wouldn't have said the things that he said. So I basically had to explain my entire paper to him over a course of several emails. And then he finally realized that he had made a mistake and that I was right. And so he said in the last email, talking about the long transcript, not the short transcript, or the long messenger RNA produced from this gene that has all 3 exons, he says, in this transcript, the fusion site is in the middle of the first exon, as you know. Well, actually, it's not in the middle of the first exon, it's in the middle of the first intron. But anyways, he acknowledged I was right. And then Casey Luskin actually took this piece from this email where Miller acknowledged I was right and published it in an article in The Blaze, which is an online media operation. And so people can actually get this article that Casey Luskin published with Ken Miller's quote from the email admitting I was right. So basically, everything is exactly as I described it, but not as I'm describing it really, but it's because that's what the data says. Yeah, one thing that's interesting about the fusion site, when they first discovered it back in 1992, now when I talk about when they first discovered the DNA evidence for it, when they sequenced what's called a DNA clone from human chromosome 2 from that region, when they found these telomere repeats, even though they were degenerate, it was not what they were looking for. They say that in the paper because they know that when fusions occur in nature, in animals that actually survive these things and sometimes even reproduce afterwards, that these fusion events always involve something called satellite DNA. So you can have, actually the PowerPoint presentation I gave you has got a picture describing it. So you can have a chromosome break and have 2 regions of satellite DNA fused to each other. You can have a telomere fused to satellite DNA, but you never see telomeres fusing to each other. In living animals, sometimes you will get telomeres that will fuse to each other in cancer cell lines, which is, I think it's safe to say that's not a healthy
No, definitely not.
living tissue. But I have evolutionists always say this. Yeah, well, look at cancer cell lines. Telomeres fuse in there. Well, that's not a healthy living tissue indicative of a healthy organism. Cancer genomes are completely rearranged and completely haywire.
Right, definitely not onwards, upwards evolution.
No, they're not. They don't represent,
Right.
functional normal cells. So anyways, we never see these things in living animals. So when they discovered this degenerate telomere region, it was not what they were looking for. They were looking for something that had satellite DNA in it. So if the human chromosome fusion was real, it would be the first documented instance of a telomere-telomere fusion ever occurring in nature.
Right.
But obviously that's not what it is.
Right, because two halves of the gene aren't going to come together from two chromosomes.
Well, telomeres are designed, actually telomeres are highly functional machines at the end of your chromosome.
Right.
They contain a very complex set of proteins that are grouped together called the shelterin protein complex that sits at the end of your chromosome, and the DNA at the end of your chromosome actually folds back into itself to form a DNA quadruplex, and then the shelterin complex sits down governing all of that and encasing all of that. And it's not just a static complex, it's actually a functional set of active proteins that also interact with elements of the cell cycle and various pathways inside your cell. So telomeres are actually functional machines, entities at the ends of your chromosomes that are doing all sorts of important things to maintain genome stability and to keep your cells functioning properly.
Right.
So they're not just these bare ends hanging out there waiting to fuse.
Right.
So they're designed not to fuse, basically. That's
Yeah.
when you think about linear chromosomes compared to the circular chromosomes in bacteria or archaea,
Right.
you have a logistical problem at the ends. You have an engineering problem that must be solved, and not really a problem, but
Right.
from our perspective of intelligent design. But if you were a human engineer, it would be a serious issue to have to deal with and to solve.
Right. Okay. Let alone blind chance.
Right.
So pesky creationists didn't shut up about this. All right. So if we can back up just for a second, because so the viewers can understand what we're talking about here. You said the fusion site produces a non-encoding RNA.
Well, this gene does not produce a protein. So there's over twice as many genes in the human genome as there are protein coding genes that produce RNAs. And these RNAs do all sorts of things. Some of them stay in the nucleus. Some of them go out into the cytoplasm. A very small percentage are even used as signaling molecules in what are called exosomes outside the cell. But a lot of these long non-coding RNAs function in the nucleus, and they modify DNA. They're called chromatin modifying RNAs. And then a lot of them also combine with proteins to form these big complexes with proteins. The RNAs sort of act as backbones for these proteins to bind onto, and they also contain regions that bind to DNA as well in the genome. In fact, there was a paper a few years back where this particular secular research paper said that the genome is like an RNA machine. So really, it's an RNA world in the nucleus of a cell in the genome. And so this gene produces long non-coding RNAs of various different types. It's alternatively spliced. It has dual promoters. It has a promoter in front of the gene. Promoters are like switches in genes that turn the genes off and on and are involved in causing these genes to produce transcripts or RNAs at certain levels and certain amounts at certain rates in the right tissues at the right time. And so it turns out that the second promoter in this particular gene, it's called the DDX11L2 gene, The second promoter in this gene is the fusion site. So this is not just DNA sitting there doing nothing. The fusion site, and I have to use the term fusion site because it's really not a fusion site. It's a promoter inside a gene. And it's very common for genes to have promoters in their first intron inside them, as well as promoters in front of the gene. This is common across the human genome. And another important thing to understand is that you have genes on both strands of the DNA molecule. DNA is double-stranded. You have a plus strand and a minus strand. When we annotate genomes, we always annotate genes as to whether they're on the plus strand or the minus strand. And the genes run in opposite directions from each other on these strands. This particular gene is on the minus strand. So the message is not your typical message that's portrayed in the fusion site sequence. It's actually in the reverse complement of that sequence. So that's what the machinery in the nucleus, the various proteins that are called transcription factors are recognizing is in this other orientation on the minus strand. And so my new research that I have developed totally confirms that this fusion site is totally functional. It binds all sorts of transcription factors, over 50 different transcription factors, including RNA polymerase II, which is the main polymerase that transcribes genes in the genome. Okay, RNA polymerase II transcribes all the protein coding genes and the long non-coding RNA genes. And in fact, these genes, these protein coding genes and long non-coding RNA genes share many of the same transcription factors. But what's really cool about this gene that contains this alleged fusion site that functions as a second promoter is that it's highly networked with many other genes that are involved in high-level cell processes, high-level, very important cell processes. And so it's heavily networked with these other genes. It's heavily integrated into the genomic networks of the cell. It functions in over 250 different types of cell types that have been analyzed so far. And what's also really interesting is that my new research, mining the new ENCODE databases and these databases, this new version of a database in Japan called the Phantom database, which is also a database that studies gene expression, shows that this fusion site region, which is about 798 bases long, contains multiple start points for transcription where RNA polymerase II binds. So there's multiple transcription start points within this region, or transcription start sites. They call them TSSs for transcription start sites. So this is a hotbed of activity. It's not a fusion site. It's a functional region inside a gene that is completely on fire and highly expressed.
Right. And just for the sake of the viewers, when you say a transcription site or a starting site, you're saying there's multiple ones, basically, when it's reading the DNA and doing something with it?
That's where the messenger RNA is starting. The 5' end of that messenger RNA, that's where it is starting at the transcription start site.
Right. So you got multiple things that are starting in that region.
Yes. Right.
Okay, and have you published on that yet?
No, I'm going to try and publish on that this year.
Okay, I look forward to that one. Now, you just came out with a paper in Journal of Creation. You co-authored with Brian Thomas, also there at ICR with you, with the provocative title, How Reliable Our Genomes from Ancient DNA? Now, this is an excellent question. You deal with the DNA that was found in what is claimed to be extremely old fossil skeletons of animals, and in particular humans. The Denisovan skeletons were one I've discussed here on the show before. Why don't you share a little bit about your paper, and why this question is so significant? Why should we care about how reliable the genomes of ancient DNA are?
Okay, well, there's actually a story behind this, that sort of my own personal story and quest is behind this. I began downloading these ancient DNA sequences from Neanderthal, from Denisovan, and I began noticing that there were some serious quality issues with these sequences. And so I wanted to really get to the bottom of what was going on. So I contacted a researcher at the Max Planck Institute, which is kind of the center of the universe for for ancient human DNA, or what they call archaic human DNA now. By the way, that's what they officially call the Denisovan and the Neanderthal specimens, is archaic humans now. That's what the people in the field call them. Yet there's all this controversy: are these humans, are these not humans, blah, blah, blah. Well, the people in the field are calling them humans. So anyhow, so I began actually corresponding with this researcher at the Max Planck Institute. They were very nice, very, very cordial. They gave me access, actually, to not only raw sequence, but also their new Neanderthal genome assembly they had not yet published.
Oh, wow. Okay.
Which I thought was very nice. And so I started by downloading the smaller chromosomes, the Y chromosome and chromosome number 22, because my computational resources at that time were not as powerful as they are now. Now we've got some very nice computational servers I have access to, and I can handle very large data sets. But at the time, I didn't have that. So I downloaded these sequences and began working with these assemblies. Now, these are what are called assemblies, where they actually took the raw Neanderthal DNA sequence, they cleaned it up, and then they assembled it into alleged chromosomes representing these ancient humans. And so I began working with these sequences, and basically, overall, my assessment was that the quality wasn't that great, but it was basically just human DNA for the most part. And then I began emailing back and forth, and then what the researcher that I was working with said, well, quit working with the Y chromosome when now we think that this is a female individual.
And only males have Y chromosomes.
And so I'm thinking, well, what's this Y chromosome doing in here? Now you think it's a female? Well, it turns out that this is not a hidden controversy. It was actually discussed in several journal papers. And their excuse was, well, we found a few genes that were similar to those on the X chromosome and accidentally misassembled this entire Y chromosome, based on these sequences they're called X degenerate genes that are on the Y chromosome. But anyways, they claim they got their X and Y chromosome sequences mixed up. Well, that really is a pretty poor excuse for assembling an entire Y chromosome and then claiming it all of a sudden it's a female and then getting rid of it. So at that point, I lost all faith in the Neanderthal sequence and the Denisovan sequence and quit working with it. And I also began studying the issue, and my co-author Brian Thomas did a lot of study on DNA kinetics and how fast DNA actually deteriorates under these types of conditions. They're finding these skeletons and these bones. And really, there really shouldn't be much DNA there to begin with, because DNA deteriorates very rapidly in just a few thousand years. Yet they're claiming, I've got the data here, they're claiming in some of these cases that these individuals are 50,000 to 300,000 years old, which is really absurd. There shouldn't be hardly any DNA there after 5,000 to 10,000 years. And of course, that fits with a biblical time frame. And based on studies and how fast DNA decomposes and falls apart, it's totally in line with a biblical timeline of just 5 to roughly about 5,000 years. I think these individuals were probably from after the flood. The DNA is probably 3,000-4,000 years old or so. I think these were individuals after the flood that were forced to live in caves and things as civilization began to be built back up. They're not found in flood sediments. They're actually buried skeletons that people died and then their relatives buried them, basically, is what it looks like for the most part. So
okay, so what about the argument then that they are 300,000 years old? This just proves that the DNA can last longer than we thought. Because we hear that one all the time when we discuss DNA and carbon-14 found in dinosaur bones, for example.
Well, everything is calibrated by evolutionary time, by the circular reasoning of paleontology, which you've probably covered numerous times on your show, so I don't need to get into that. Literally every field that there is in science is calibrated by that. And so that's what's going on here in this situation. And one thing I want to mention before I forget is that when I began studying this issue in even more detail, I realized that the amount of modern human DNA contamination in these samples is really high and a really big problem. So that Y chromosome that was in that assembly was probably from modern humans that were handling the specimens, and their DNA was sequenced. And it turns out that's a big problem. There was a paper published in 2009 or 2010 where researchers actually went into all of these non-primate genomic assemblies and DNA databases, and many of them had up to 10% human DNA contamination in them. In fact, there was an entire region of the zebrafish genome that was fully human because it was human DNA contamination that was assembled into the zebrafish genome assembly.
Oh dear.
Yeah. And so what does that tell us also about all these primate genomes that are being sequenced? How much of them is just really human DNA and not ape DNA? And of course, it's really bad in these Neanderthal and Denisovan genomes. You just can't tell the difference. These are archaic humans. How do you tell what the difference is between a lab worker's DNA and what you're getting that's actually real archaic DNA? It's just nearly impossible to tell. There are different ways that they use to try and filter that contamination out, and they claim now that they can get only 2 to 5% contamination in a modern new genome assembly, but I'm pretty skeptical about that.
Right, and even then, that's still 2 to 5%. That's still a pretty significant amount. That's
still pretty significant. The problem is that archaic DNA is cool. It's sexy. It gets you big grants. It makes you a rock star in the academic world. It gives you a niche to fill and to be a rock star in, basically, and to make a career out of. And I think that's really is a lot of what's going on in this area. Now, I'm not saying that there's not valid research to be done there, but I think we need to be very careful about these genomes and these genome assemblies. And you made some interesting comments as well about comparing human genomes from Africa and the diversity there compared to these archaic humans.
Yeah, and that had to do with, you brought up the Denisovan DNA sequencing, because when that paper came out, literally the same week, another paper came out in Cell magazine where they sampled the DNA of hunter-gatherer populations like the Pygmies, the Sundawes, the Hadza tribes.
Sure.
These are groups of people still alive today. In the Denisovan paper, Meyer and team were trying to show how these Denisovans were so different from modern humans, apparently to make them look like our ancient subhuman ancestors. They were talking about some 111,000 differences in the DNA, yet Lachance and team identified more differences in the DNA between the modern human baseline and the Pygmy and Sandawe tribes. These are completely modern humans. I mean, they're alive today. They can have children with other humans around the globe. So from these differences in the DNA, speculation springs up from, with the researchers trying to put a date on when the Denisovans and humans branched off from one another. They claimed between 170,000 and 700,000 years ago, again, well beyond the biblical time frame. But this age was based upon these differences between the Denisovan DNA and the modern human DNA. Differences that were smaller than the differences between modern humans still alive today. So that argument is bankrupt. And now Thomas and yourself are pointing out that DNA can't last that long anyway. I mean, the Denisovan bones were found in a damp cave. The conditions for preserving DNA do not get any worse than that.
So they're typically within the range of modern human DNA variation. Absolutely. Well, that's what these people call these remains. They call them archaic humans now. At least the people in the DNA field, that's what they do. Now, I know there's arguments in these other areas where they don't necessarily study that specifically, but these are basically old, ancient humans.
And of course, the whole point I was trying to bring up, which you guys brought up in your paper, was they were talking about 170 to 700,000 years ago, well outside of the biblical time frame. A huge point of your paper with Brian Thomas was these ages are based in circular reasoning. The evolutionary age, in this case 170 to 700,000 years, is wrong because they base it upon that and say, isn't it amazing the DNA lasted this long? Now we come to the Denisovans, we're talking about these skeletons were sitting in damp caves. It's the worst possible conditions for preserving DNA. I mean, it just doesn't get any worse than that.
We know how fast DNA decays. We know how fast it decays under certain temperatures, under certain moisture conditions. And it's just literally impossible for these remains to produce viable DNA after more than about 5,000 years, 5,000 to 7,000 years. 10,000 max under really good conditions.
And so that would probably be a pretty safe date to place upon, say, the Denisovan and Neanderthal skeletons, simply because we can extract DNA from them. And it decays so fast, it's pretty solid to say that they can't be any more than 10,000 years old.
That's pretty safe.
All right. Okay, well, thanks so much for taking the time to be on the show today. I know the viewers appreciate it.
All right, thank you.
Dr. Tomkins and ICR have an excellent new video series out entitled Unlocking the Mysteries of Genesis, which of course includes lectures from Dr. Tomkins and Brian Thomas, as well as many others. You can get your own copy at unlockingthemysteriesofgenesis.org. This interview was heavily edited for television. You can catch the entire meaty interview on our YouTube channel. Just head on over to genesisweek.com, which will take you to our YouTube channel, and you can watch the entire interview, which was pushing 40 minutes long. You can also subscribe to our YouTube channel while you're there. Stick around. We'll be back in just 1 minute.